Good morning, students. Now we will start protein metabolism. We have already completed chemistry of amino acids and proteins in competition number 5.1 and 5.2. Now competition number 5.3 and 5.4 is of metabolism. So out of this, today we will study this competition number 5.3, and today's topic is digestion and absorption of dietary proteins. Daily dietary protein in our diet ranges from 50 to 100 grams per day. Digestion and absorption of protein is a very efficient in healthy humans, and hence very little protein, that is 5 to 10 grams per day, is lost to the feces as an undigested proteins. Coming to the definition of digestion, first we will see digestion, then we will see absorption. Digestion is the disintegration of complex nutrient into simple, soluble and absorbable form, or simply you can say digestion means conversion of complex unabsorbable food into simple absorbable food is called as digestion. Major source of nitrogen is the protein which we consume in the diet. Proteins are too large to be absorbed as it is, because protein is a polymer. Our body absorbs monomers only. Like starch is a polymer, it is not absorbed, but when it is converted into glucose, which is a monomer, then it is absorbed. Similarly, protein is a polymer of amino acid. So proteins are not absorbed from our alimentary canal, instead of which these proteins are first digested and converted into various amino acids, and then these amino acids are absorbed. This protein digestion requires various hydrolytic enzymes, and they causes breakdown of protein by a process called as hydrolysis. After breakdown, proteins are converted into amino acids. So these proteolytic enzymes are present in stomach, pancreas and small intestine. This proteolytic enzyme belongs to the hydrolase class of enzyme classification, that is class 3. These proteolytic enzymes are secreted in the inactive form and they are synthesized in inactive form and stored in the cells of alimentary canal in the form of zymogen. When they secreted into the small intestine, they becomes active. The active site of this enzyme, when they are stored, are masked by a small region of peptide chain which removes by hydrolysis of a specific peptide bond. That means when these zymogens are stored in the intestinal cells, epithelial cells, their active site is masked due to presence of a small polypeptide chain over it. And when they are secreted into the intestinal lumen during digestion, that flap which masks the active site is removed and active site is now open. And when this active site is exposed, this enzyme becomes active. These proteolytic enzymes may be exopeptidase and endopeptidase. Exopeptidase means the enzymes which break the proteins from their ends. All of us know that proteins are nothing but they are polypeptide chain which have two ends, carboxyl end and amino terminal end, C terminal and N terminal. So exopeptidase will remove the amino acid either from carboxyl end or from amino terminal end, whereas endopeptidase will break the polypeptide chain from inside. Now proteolytic enzymes are secreted by stomach, pancreas and small intestine. Stomach secretes two proteolytic enzymes, pepsinogen and renin, whereas pancreas secretes trypsinogen, chymotrypsinogen, elastase, carboxypeptidase A and B. Small intestine secretes aminopeptidase, dipeptidase and enterokinase. Now we start actual process of digestion. So digestion in oral cavity. Oral cavity has saliva, but there is no proteolytic enzyme present in the saliva. So practically there is no digestion of proteins in the saliva. But carbohydrate digestion takes place in the oral cavity due to presence of salivary amylase enzyme in the saliva. So when there is no digestion in the saliva, in the mouth, but saliva makes the food lubrication. Saliva also helps food making soluble for the action of proteolytic enzyme. Now, after mastication, the food enters into the stomach. Stomach contains gastric juice. This gastric juice contains hydrochloric acid and two proteolytic enzymes, that is pepsin and renin. First, we will see what is the role of hydrochloric acid. Hydrochloric acid causes denaturation of the proteins, and all of us know when proteins undergoes denaturation, its secondary, tertiary, quaternary structure is disturbed and the size of protein increases, and due to which the proteolytic enzyme can easily act on such proteins and easy digestion of protein takes place. Therefore, we can see that boiled egg is easily digested as compared to raw egg. So, HCl causes denaturation of protein. At the same time, HCl also leads to activation of some proteolytic enzymes. Just in the previous slide, we have studied that proteolytic enzymes are present in inactive form. So, some of the proteolytic enzymes are activated by HCl. After studying the role of HCl, we will study now the role of proteolytic enzyme. The first proteolytic enzyme is pepsinogen. This pepsinogen is inactive. HCl act on this pepsinogen and convert it into pepsin. Once this pepsin is formed, which is the active proteolytic enzyme, it further acts on inactive pepsinogen and convert it into active pepsin. This conversion is called as autocatalysis. Now this pepsin act as a proteolytic enzyme. It will break the protein, or it will causes the digestion of protein, and for this it requires a pH between 1.6 to 2.5, and this pH is provided by HCl. This pepsin becomes inactive if the pH is above 5. Now pepsin breaks the peptide bonds where carboxyl group is contributed by aromatic amino acid like phenylalanine, tyrosine and tryptophan. All of us know that peptide bond is formed between two amino acids. One amino acid will contribute carboxyl group and another amino acid contribute amino group, which is shown here. On dehydration, COH group and NH2 group get converted to CONH group, and CONH group is called as peptide bond. Now in this example, this carboxyl group is given by tyrosine, TY stands for tyrosine. So pepsin will break those peptide bonds in which carboxyl group is contributed by aromatic acid, amino acid like phenylalanine, tyrosine and tryptophan. So this peptide bond can be broken down by pepsin because its carboxyl group is given by tyrosine. Apart from tyrosine, it will also break the peptide bonds in which carboxyl group is denoted by phenylalanine and tryptophan. This pepsin has strong action on milk protein like casein, and it hydrolyzes the protein into small pieces called as peptides. Next enzyme is renin. Renin is secreted in infant, but it is absent in adult gastric juice. It is secreted in the form of prorenin, and this prorenin is converted into renin by HCl. Something about renin. This renin causes digestion of proteins, for which it requires optimum pH 4. The pH of infant protein is higher as compared to adult, so it can work there. It also acts on a protein called casein, which is present in the milk, and here this renin act on casein, converted into para-casein. This para-casein combines with calcium ion to form calcium para-caseinate. When there is deficiency of this renin in the infant, then renate tablets are now available in the market, or they are also used in making the cheese. All proteolytic now digestion in small intestine by pancreatic juice. All pancreatic enzymes are active only in alkaline medium. So this alkaline pH is provided by bile juice, which is synthesized by liver, and this bile juice contain bicarbonate ions which provide the alkaline medium. The proteolytic enzyme present in the pancreatic juice are trypsinogen, chymotrypsinogen, proelastase, collagenase, procarboxypeptidase A and B. First we will see trypsin. This trypsin is secreted again in inactive zymogen form, which is called as trypsinogen. The enzyme enterokinase, this enterokinase enzyme is secreted by intestine. It is present in intestinal juice, not in the pancreatic juice. Now this enterokinase act on trypsinogen and convert it into trypsin. Once this trypsin is formed, this trypsin then acts on more inactive trypsinogen and convert them into active trypsin. Again, it is an example of autocatalysis. Trypsin is an endopeptidase and it will break the peptide bonds in which carboxyl group belongs to basic amino acids like arginine and lysine. In this example, I have shown one peptide bond CONH, where the carboxyl group of this peptide bond is denoted by arginine. AR means arginine. Instead of arginine, if lysine is there, then also that peptide bond is also broken down by the trypsin. So these proteolytic enzymes are group specific enzymes. They are not bond specific. They will not break all peptide bonds. They break specific peptide bonds. Trypsin break the protein into peptides. Trypsin is also required for conversion of another proteolytic enzyme, that is trypsinogen into chymotrypsin, chymotrypsinogen into chymotrypsin, and proelastase into elastase. Next, role of chymotrypsin. Again, it is secreted in the form of zymogen, that is chymotrypsinogen. Trypsin act on it and convert it into active form chymotrypsin. It is an endopeptidase and break the peptide bonds of aromatic amino acids like phenylalanine, tyrosine and tryptophan present on the C-terminal end. So here, if you see this example, at the C-terminal end of the polypeptide chain, the phenylalanine is present, and that phenylalanine forms a peptide bond with adjacent amino acid. Such peptide bonds in which phenylalanine, tyrosine and tryptophan are involved are broken down by this chymotrypsin. This chymotrypsin also breaks the peptide bond formed by leucine, methionine, histidine and asparagine. Next, role of elastase. Again, it is secreted in inactive form called proelastase in pancreatic juice. Trypsin act on proelastase and convert it into elastase. It is endopeptidase which cleaves the peptide bond in which carboxyl group is contributed by glycine, alanine and serine. So this is the example of peptide bond, CONH bond, where the carboxyl group is contributed by glycine. Instead of glycine, if alanine and serine is also if present, that type of peptide bond is also broken down by elastase. Next, procarboxypeptidase. There are two procarboxypeptidase, A and B, and they will convert it into carboxypeptidase by the action of trypsin. So first, carboxypeptidase A. It is a metalloenzyme contains zinc. It is a exopeptidase, means it will not break the peptide bond in the middle, but it break the peptide bond from the periphery. Again, it is a group specific proteolytic enzyme where it acts on those peptide bonds in which carboxyl group is contributed by hydrophobic amino acids like alanine, leucine, valine and isoleucine. Here I have given some example. In this example, the peptide bond is formed shown as CONH, and this CO group is contributed by valine. So instead of valine, if alanine, leucine and isoleucine is there, such type of peptide bonds are also broken down or hydrolyzed by carboxypeptidase A. Coming to carboxypeptidase B, it is also exopeptidase. It acts on peptide bond of which carboxyl group is contributed by either arginine or lysine. Same. All these proteolytic enzymes break the protein into small peptides. After studying pancreatic juice, now we'll see the digestion by intestinal juice. Intestinal juice contain aminopeptidase, dipeptidase and enterokinase. Aminopeptidase, as the name suggests, they are exopeptidase and they break the amino acids from amino terminal end. They are leucine peptidase, they require magnesium or manganese for their activity. They are exopeptidase, and as I have told you, they act on the amino terminal end of the protein and releases the amino acid. Dipeptidase, they act on small dipeptides which is made up of two amino acids bound together by single peptide bond. These dipeptides contain cobalt or manganese. They act on dipeptides and break the dipeptide into free amino acid. Enterokinase, if you remember, initially I have told you this enterokinase is the enzyme which convert inactive trypsinogen into trypsin, and this trypsinogen is present in the pancreatic juice. After activation, this trypsin will causes autocatalysis as well as it will activates the other inactive zymogen enzymes present in the pancreatic juice. Now this is about digestion. Now coming to absorption. After the digestion, what we get is we will get large amount of amino acids, but some oligopeptides are also found. So the product of digestion are oligopeptides and amino acids. Site of absorption. Oligopeptides are absorbed from duodenum and proximal jejunum inside the cell, and in the epithelial cell they get hydrolyzed into amino acids, whereas amino acids are directly absorbed into the blood from distal jejunum and ileum. So these oligopeptides are absorbed taken up by the epithelial cells, and inside the epithelial cells they are further broken down into free amino acids. Our dietary proteins mainly contain L amino acids in large quantity. So these L amino acids are actively transported with the help of carrier. So active transport means they require energy from ATP. And the carrier, these carriers are very specific, means 5 different types of carriers are present for transport of various different types of amino acids, and all these carriers depend on sodium, therefore they are called as sodium dependent carriers. D amino acids are not absorbed by simple diffusion. So these are the 5 different types of carriers. First, for absorption of small and neutral amino acids, this carrier will absorb alanine, serine and threonine only. Second type of carrier is for absorption of large neutral amino acids like valine, leucine, isoleucine, phenylalanine, tyrosine, tryptophan. Third type of carrier is for basic amino acids like arginine, lysine, ornithine, cysteine. Fourth type of carrier is for absorption of acidic amino acid like glutamic acid and aspartic acid. And fifth type of carrier is for absorption of amino acids and glycine like protein, hydroxyprotein and glycine. So this is the absorption. Here three compartments are there. On the left I have shown you intestinal lumen, in the middle intestinal epithelial cell, on the right side it is a blood. In the left side I have shown a carrier which has two sides. At one side amino acid binds and at another side sodium binds. When both these sides are full, this carrier will rotate to 180 degree and now it faces in the intestinal cell. Along with that, amino acids and sodium also enter inside the intestinal epithelial cell and they get released in the intestinal epithelial cell. Later on, the amino acid is directly absorbed into the blood. Now sodium, which has entered with the help of carrier in the intestinal cell, sodium is a extracellular ion. So cell want to expel sodium outside, but in the extracellular space the sodium concentration is more. Therefore cell has to expel this sodium against the concentration gradient. Therefore it requires a pump called sodium potassium ATPase or sodium potassium pump. This sodium potassium pump will then expel the sodium from intestinal epithelial cell to extracellular space against the concentration, so it requires ATP. So in this way one ATP is utilized for absorption of amino acids indirectly. Therefore it is called as active type of transport. So this type of transport is seen in intestine. One more type of absorption transport is there, which is called as Meister cycle or gamma glutamine cycle. Here by this cycle the amino acids are absorbed not only in small intestine but in renal tubules and brain. So this is the diagram of Meister cycle. If you see here, the cell membrane is shown. One amino acid is present above the cell membrane, that means outside the cell membrane, and inside the cell membrane an enzyme called transferase is present in the cell membrane, and in the cytoplasma a glutathione is present. Glutathione is nothing but it is a tripeptide which is made up of glutamic acid, cysteine and glycine. Now what will happen when amino acid come close to the cell membrane? The enzyme transferase will break the glutathione which is present inside the cell and take the glutamic acid from glutathione and combine that glutamic acid with the amino acid which is present outside, and the combination of amino acid and glutamic acid is then absorbed with the help of enzyme gamma glutamyl transferase. Now this glutamic acid and now amino acid will enter inside the cell. Now this combination is immediately broken down. The amino acid becomes free and the glutamic acid also becomes free. Now the glutamic acid, which is a straight chain structure, immediately get converted into oxoproline, a cyclic structure. Oxoproline is a cyclic form of glutamic acid. With the help of enzyme, second enzyme, that is gamma glutamyl cyclotransferase. Right. Now this cyclotransferase is converted back into glutamic acid by a third enzyme called as oxoprolinase, which break the cyclic structure with the help of ATP, and glutamic acid is now formed. Now initially when glutamic acid is removed from glutathione, cysteine and glycine, they get separated. Now when this glutamic acid is reformed, this glutamic acid combines with the cysteine once again with the help of fourth enzyme called as glutamyl cysteine synthase to form glutamyl cysteine. For this combination or for this formation, again energy is required, which is supplied by ATP. Now then this glutamyl cysteine combines with their third friend, that is glycine, with the help of fifth enzyme called glutathione synthase. Again it requires energy, which is supplied by ATP, and glutathione is regenerated. So in this way the Meister cycle helps in the absorption of amino acids. Some important thing about the absorption: generally proteins are not absorbed, but in infant for a short period of time after birth, small intestine can absorb some intact proteins by the process of endocytosis or pinocytosis. And the example of this is absorption of immunoglobulin A, which is present in the colostrum of mother milk. They are absorbed as it is, and this immunoglobulin A protect the infant from various diseases. In an adult intestine, intact proteins are generally not absorbed. But sometimes if there is absorption of intact protein in adults, then it is responsible for formation of antibodies. Means when that protein, dietary protein, enters in our body as it is, then it acts as an antigen, and our body will synthesize antibodies to such protein, which will leads to various types of food allergy. The most common type of food allergy is gluten induced enteropathy. What is gluten? Gluten is a protein present in the wheat, and many individuals have allergy for this gluten. Second, fish allergies, egg allergies, all these are examples of food allergies. So we have completed digestion, absorption and transport of dietary protein. Now we'll see in short about disorders like Hartnup disease, cystinuria and glycinuria, which are related to digestion and absorption of proteins. First inborn error in digestion and absorption of amino acid is Hartnup disease. The name originates after the first family in whom the disease was described. It is an inherited autosomal recessive disease. Here the absorption of aromatic amino acids from intestine as well as reabsorption of these aromatic amino acids from renal tubules are defective. So these amino acids are excreted in the urine. That means the aromatic amino acids present in the small intestine are not absorbed. At the same time, the aromatic amino acids which are filtered through the glomerulus, they are also not absorbed back. Now, generally, the amino acids which are filtered through the glomerulus are reabsorbed by the proximal tubule. But in such cases, these amino acids are not absorbed or reabsorbed. Now, patient with Hartnup disease have pellagra-like symptoms. Now, what is pellagra? Pellagra is a disease which is due to deficiency of niacin, vitamin B3. Now, how will you correlate this? For this purpose, you should know this slide. Here, if you see, tryptophan have various fats, or it can be converted into various substances, and one of the compound is NAD+. So this NAD+ is a coenzyme in various reaction, and this NAD is synthesized from niacin. And if tryptophan is not absorbed in Hartnup disease, in our body 60 gram of tryptophan is converted into 1 milligram of niacin. If tryptophan is not absorbed, then niacin is not synthesized in the body, and when this niacin is inadequate in the diet, then such person develops pellagra disease, and it will lead to deficiency of NAD, which is a coenzyme. The patients with heart nerve disease have common manifestations like dermatitis and ataxia. The neuropsychiatric manifestations of heart nerve disease are first observed in the behavior of emperors like Nero and Caligula, because they are also suffering from the same disease. Now, how do you diagnose this heart nerve disease? Amino aciduria is there. Here there is increased excretion of indole compounds. Indole compounds means indole is a group which is present in tryptophan. So indole compounds are, as the tryptophan is not absorbed, it is excreted into the urine, and that indole compounds are detected by Obermeyer test. Treatment includes high protein diet, supplementation of niacin, and minimum exposure to sunlight, because dermatitis is also seen in such patient. Next is cystinuria. It is also an autosomal recessive condition. Here again there is defect in the transport of cysteine, and along with cysteine other amino acids like ornithine, arginine and lysine are also not absorbed, again not from intestine as well as from the renal tubules. So cystinuria is one of the inborn error of metabolism associated with Garrett's tetrad. Garrett's tetrads means 4 tetrad, as the name suggests, 4 different conditions are there: alkaptonuria, albinism, cystinuria and pentosuria. In alkaptonuria, alkapton bodies are excreted into the urine. In albinism, melanin is not synthesized. In cystinuria, cysteine is excreted into the urine. And in pentosuria, pentose sugars like ribose, ribulose, xylulose, they are excreted into the urine. So these 4 conditions are generally associated with each other, and then it is called as Garrett's tetrad. So whenever there is a cystinuria, that is cysteine is excreted into the urine, also at the same time ornithine, arginine and lysine are also not absorbed. Now, signs and symptoms includes abnormal excretion of 4 amino acids like cysteine, ornithine, arginine, lysine in the urine. Crystalluria. In acidic pH, these cysteine crystals are formed in the urine. If the pH of urine is acidic, so cysteine amino acids will accumulate and form say crystals. These crystals may lead to renal calculi or renal stones, and these renal stones sometimes obstruct the urinary blood flow and may lead to renal insufficiency. Now, treatment of this condition includes increase urine volume by taking more and more fluid, so that chances of crystal formation are less. And then we have to solubilize this cysteine crystal by alkalinization of the urine or by giving sodium bicarbonate. What we have seen in acidic pH, the formation of cysteine crystal, chances of formation of cysteine crystals are more. Therefore, if we make the urine alkaline by giving sodium bicarbonate, these crystals get dissolved. Last is glycinuria. It is also a genetic condition where defect in the transport of glycine from intestine, again with renal tubules. So here also glycine from small intestine is not absorbed. At the same time, the glycine from renal tubule is also not absorbed. Therefore, that glycine will excreted into the urine, where it forms calcium oxalate stones. And how will you diagnose this? You can diagnose this by excretion of large quantity of glycine in the urine. Here, management of again kidney stones. If you see these 3 conditions, heart nerve disease, cystinuria and glycinuria. In cystinuria and glycinuria, when these amino acids are not absorbed from the GIT and renal tubules, our body has no effect because they are non-essential amino acid and body will synthesize these amino acids. But due to excretion of, because due to non-absorption from renal tubules and excretion into the urine, these cysteine and glycine forms renal stones. So non-absorption from diet of cysteine and glycine does not affect our body, or excretion of cysteine and glycine into the stool does not affect our body because they are non-essential amino acid and body can synthesize them. But due to non-absorption from renal tubules of cysteine and glycine, they leads to various crystal formation. Now, other defect in the digestion and absorption of amino acids are also seen in partial gastrectomy, means half of the stomach is removed. If patient is having pancreatic disease, so pancreatic proteolytic enzymes are not synthesized in proper quantity, and one more condition is called as protein losing enteropathy. So these are some conditions in which there is defect in the absorption of amino acid is seen. So this completes your digestion and absorption of proteins and amino acids. Thank you very much.